All right, well, welcome to the second part of the Spring 2021 Undergraduate Research Workshop Series, Developing Research Skills. So this is a series of workshops that we are collaborating with the Merit Scholars Program, as well as some wonderful graduate students here who are helping us produce and put on these great workshops on developing these skills. So these are kind of skills that we see as things that are the second tier. You know, you've already got kind of a foundation of knowledge. Maybe you just started doing research, and now you're starting to think about, doing lit reviews and, analyzing different data journals and all those sorts of things. So this week we're very excited to have a brand new workshop called Scientific Literacy, How to Read a Paper. And we're very excited to have all of our wonderful presenters. But I'm going to hand over the time. And once again, my name is Gareth Jones. You can probably see it there on the recording. But I'm the Assistant Director in the Office of Service Learning and Undergraduate Research. We also have some of our wonderful Vistas in our office who are here. You can contact any of us at any time, and we are happy to help you with finding opportunities, to answering any type of questions that you have about undergraduate research and service learning. That's what we're here for. But from this point forward, I'm going to go ahead and hand the time now over to Keegan Curry, who will introduce himself, and then we'll introduce our wonderful presenter.
Yeah, thank you for that, Gareth. So my name's Keegan Curry, and my friend Haley Levi and I are first-year PhD students here at UAB. And we just had an idea for this workshop series to really just help undergraduate students learn research skills, because in a lot of ways that really isn't covered in the standard curriculum these days. And so I'm going to hand it off to Casey McPherson, and she put together a wonderful talk for everyone. So.
All right, just a little bit about myself. So I got my PhD in medical pharmacology at the University of Mississippi Medical Center, and I traveled right down I-20 to UAB. I have been at UAB as a postdoctoral fellow for 3 years, but I just recently this summer got accepted into the Merit Fellowship Program. I am in Dr. Brittany Lasane's lab in the CETA department at UAB. And right now I'm basically doing data science with kidney disease, just in a catch-all phrase kind of thing. And today I'm going to be talking about scientific literacy, how to read a paper. Today we're going to go over the sources of information. So just a brief introduction of where information comes from and the different types. We're going through lit search strategies and how to use PubMed and other alternatives. The third section will be talking about info quality check and evaluating the quality of the information you have found based on source and content, how to read these papers once you find them, and how to store that information once you got it. All right, so for sources of information, we're going to talk about the different types. Information, basically, like most things in life, has its own cycle. In order to understand, locate, and evaluate information, it's important to understand where in the cycle the information was obtained and the characteristics of each type of resources. These resources can be classified based on the originality of the material, as well as the proximity to the original material. So in other words, are you getting that information directly from the mouth, or is it like through the grapevine, if you will? Based on these classifications, we have three types of resources. We have primary resources, secondary resources, and tertiary resources. And so to understand what these different resources are and where they fall in the cycle, I have the cycle here laid out. It starts with topic selection. Can y'all see my cursor? I'm gonna see if y'all can. All right, so on the very top where it says topic selection, that's where people start off with their idea. They do research. For me, that would be in a wet lab, dry lab, collecting data from animals, from human samples, analyzing it, and taking that information and applying it or talking about it informally in lab meetings and department seminars and whatnot. After it goes through that phase, you will end up actually talking about the information that you found at conferences and presentations. And then finally, you get to the primary resource, which is the journal research. And this is the original new information from research. So this is the direct information, this is your primary source. After the primary sources have been made, they can go to the next phase where there are secondary resources. Secondary resources summarize and synthesize information from the primary. These can happen as review articles, even just for both experts and non-experts. And then of course, you also have magazines and news articles as well that fall under this classification. And then finally, the one that everybody's probably really familiar with because we've all been in school for forever and a day, is the tertiary resources. The tertiary resources condense information based on primary and secondary resources. So your textbooks, your encyclopedia, Wikipedia, and whatnot. So what I really want to focus on out of those is talking about the primary and secondary resources. And there is a pretty easy way to decipher which is what. First, for primary resources, there we go. It is made of a title, abstract, introduction, method, results, discussion, conclusions, and references. These are all the different sections of your primary resources, and they all serve its own purpose. The title is just supposed to give you an overview of what that paper is about, and probably will also include some technical jargon. The abstract is just supposed to give you a short summary and like the take-home message of the paper. The introduction just gives the background, the rationale, and the purpose of the paper. The methods, as you can guess, talks about where it was done, how it was done. Results give just the results, not the interpretation, just what has happened. And then finally, in discussion, though, the interpretation of results occurs. And finally, it concludes wrapping everything up, giving a good summary. And then, of course, you have your reference cited. Similarly, secondary resources also have a title and an abstract. They also have an introduction that they all have the same function as they do in the primary resources. However, you'll notice that here, methods are typically not part of a review, not always. Certain reviews, such as systematic reviews or whatnot, you actually will have a methods section describing how you obtain the primary resources. If you did any statistics on your own on the primary resources, you would explain how you did that. But for the most part, you don't really have a method for a secondary source, such as a review paper. It'll just have topics that just kind of give a synthesized review of the literature organized in a particular way. And then finally, it'll conclude giving the summary of the main points. A lot of times people will also include future perspectives and in the context of maybe how it relates into their field, and of course, references. Before I go on to the next section, does anybody have any questions? Woohoo! All right. The next thing we're going to do is talk about literature search strategies, where and how to find that information. Okay, so the one that I'm really going to talk about is PubMed. There's several different databases, Medline, Google Scholar, I mean, tons and tons and tons. PubMed tends to be the one that most people are familiar with. It is a search engine, excuse me, for scientific literature in the fields of medicine, nursing, dentistry, all of your healthcare sciences, if you will. It contains more than 26 million references dating back all the way to 1946, and it can be found on the NCBI website. And so in order to access it, you go to this link here, and it'll pull up this page that you see here on the right. This is just the NCBI's website. This is not just PubMed. To set up PubMed research, you have to go to the drop menu called All Databases, and it will drop down and you can select PubMed. All of these, ClinVar, Geo, AMM, all of these are all different databases depending on what your field is, which one you're going to use. I probably haven't used half of these yet. I usually stick with PubMed, Geo, ClinVar. I'll even go through their books and whatnot, but today we're going to talk about PubMed. And so after you get PubMed pulled up, you just type in keywords like, so for instance, kidney disease, and right now I'm focusing on polycystic kidney disease. So I just put that in the search engine. This is the first thing that pops up. A couple of things I want to point out is here on the left, you have your results by years. You can take this little circle and you can drag it back and forth and just pick a particular time window if you'd like. You have different text availabilities. I don't like to mess with this. If there isn't a full text available, there are other alternatives on how to get that, and we'll talk about that in a little bit. Article types, it depends on why you're searching for what you're searching for. If you're searching for just for your general knowledge, I usually like to click reviews and just get the reviews on the topic. If you are specifically looking for clinical trials, you're looking for human data, human studies, you would click on clinical trials. You can do publication dates down here as well, but I typically don't mess with this part. I stick with up here with the timeline. I think that is it. Oh, up here on the right, I wanted to point out that you can sort by different ways. You can do by most recent, you can do by most closely related to what you searched for, you can do it by author, you can do it by journals. I usually stick with most recent, but you can actually click the display options here and pick which one, how you wanted the results ordered. And then you can also go to additional filters, which is this button down here. When you click that, another window pops up. And or actually, this is the advanced window. So anyway, the advanced window here, you can actually type in. So if you know, if you're looking for a particular paper or particular topics by one person, and you want all of their papers for this one topic, you can actually come to the advanced search settings. In the all fields, this is a drop-down menu where you can search by anything, last author name, first author name, the PMID number, the title, the DOI number, and many, many, many more. You can, so for instance, if I wanted to look up all of my mentors' papers, I would put in last name, and in the search bar right here, I would type in Lassane, and I would click add. When you click add, that information is going to pop up in this query. Once you're done adding all the information that you want to look for, you then push search, and it'll post all the papers that fall under those categories. Now, a caveat is if you have too many filters, it's probably going to be way too much. It may not show papers, so you'll have to dial it back some. Another thing I'd like to point out here is where it says add right here, you can also select or. So instead of saying you want Lassane and kidney disease and UAB, you can say I want Lassane and kidney disease or UAB. And at the UAB part, it won't have to be, it's not all of her papers about PKD that was just done at UAB. It would have pulled up all of her papers. And so that's ways you can play with that. And I do invite people to just play around with it, get familiar with it. This is actually really handy dandy, especially when you have homework assignments. I usually come here and start searching for particular things that fit the assignment. All right, so let's say you picked a paper. I just picked this one at random. All the information that shows up when you select a paper is the journal source information, the title of the article, the language that it was published in, if it is not English, the names of the authors, which are usually shown here as a hyperlink, the author affiliations. So that's right here. Typically, it's not expanded. You can click expand, and it'll tell you what university, what country they're, that the research was, or what the author is from. You can also do, it'll also have the abstract, the publication type, whether it's a review article or a descriptive article. And it'll also even have some vocabulary terms associated with it as well. But there's one little tiny thing. A lot of times, if you want to get the full text, you would come over here where this red box is, and you would click it. And about 50% of the time when you click it, it's going to tell you that you need to pay like $50 to read this one paper that your teacher assigned you to. And there's nothing more aggravating than a paywall. And that is a soapbox that I could stand on later for y'all if y'all need it. I won't bore y'all too much. But there are ways around this paywall, and that's what we're going to discuss next. How do we bypass that paywall? Well, if you are at a university, you technically already have that bypass. You, for instance, at UAB, go to library.uab.edu. When you go to that page, below it will be databases, and you can click on that, and it'll ask you for your login information. And once you do that, whatever database that you want, whether it's PubMed, Medline, whatever, you click on it, and all the paywalls are gone, just magically. If you don't have university access, or if for some reason the university usually may not always have access to a certain article, you can go to the ResearchGate community. So you go to researchgate.net. That is just a social networking site for scientists and researchers just to share papers, questions, answers, find collaborators. I really like it. It's pretty legit. You can actually search for a paper by title in their search engine. And if somebody has already posted a PDF of it, it'll pull it up. If there's not a PDF of it, you can actually contact the first author or last author for a copy. That's totally okay. Obviously, do it very respectfully, don't demand it, but ask them, say, hey, I'm really interested in this because blah, blah, blah. I don't have access to it through my university. Would you mind sending me a PDF of it? Most of the time they will. However, not everybody's on ResearchGate. So you can actually go to Twitter, and what I call science Twitter. It's just regular Twitter, but it's just where all the science people are too now. You can search by author names if they have an account. Always check to see how active they are on it, because if it's not very active, then most likely they won't respond to a direct message. But if it looks like they're pretty active, you can actually direct message them as well on Twitter for a copy of the paper. That's totally fine. Then lastly, if none of that worked, all papers usually come with an appointed contact person. It's one of the authors. Most of the time it's the senior author. The contact would be their address, sometimes their phone number, like their university office phone number, and majority of the times they will have their email. You can email the author and ask them, can you have a copy of the paper? All right, so once we find all of the papers, let's see. Oh, there we go. I wanted to check to see if anybody had any questions in the comments. We got to check the quality of the paper. And how do we do that? Well, there's a few ways. First thing that you can do is you can look at the major database indexing. This is how you evaluate journals, and I should back up. So to assess the quality of the information, you want to look at the journal, and then you want to look at the paper. The first thing we're doing, you want to look at the journal. To evaluate journals, the journal should be indexed in a major database such as PubMed, at least one. If it's not on PubMed, it may be on Medline, it may be on anything else, but it has to be on at least one to be a good journal. Journal publication rates, how often do they publish and how many issues do they put out? Journals should publish a certain amount of content regularly and put out new issues. You can look at the table of contents of a current and last volumes of the journal issues and get an idea of how many issues they put out per year. You can get an idea of how many papers a journal will publish in an average issue. Journals with regular readership will typically publish 10 items or more per issue and put out at least 4 issues a year, so every quarter. Another thing that a good journal should have is a really good, clearly defined editorial board that has a chief editor, and members of the editorial board should either be from the full university professors or other senior scholars from a research institute. Another thing you can look at on journal websites, you can go check out their editorial scope. The editorial scope should be clearly detailed, including the overview of covered subjects. And this is usually under their about or scope page on their website. Now, one thing that everybody tends to evaluate journals on is their impact factor. Some people say you shouldn't, but that's kind of how we do it. So we're going to talk about it. The impact factor is just how frequently on average an article gets cited from a journal in a particular year. It's used to measure the importance or rank of a journal by calculating the times the article has been cited. It's usually based on a 2-year period, though I have seen, I think some of them may do a 4-year period. It involves dividing the number of times the article was cited by the number of articles that are citable. And the way you read that is if you have an impact factor of 1, that means on average the articles published 1 or 2 years ago have only been cited 1 time. So it should be easy or clear to see that the higher your impact factor, the more legit the journal should be. Okay, so now that we've evaluated journals, let's talk about evaluating articles. The way we do that is we ask ourselves, is it CRAP? And CRAP, C-R-A-A-P, stands for currency, relevance, authority, accuracy, and purpose. And we're going to go through each of these. Currency just means the timeliness of the information. So in other words, what's the publication date? Has the information been revised or updated? Sometimes that happens. And if it does, index services like PubMed will actually have a notification near the title of the paper when you find it on PubMed saying that it has been revised or updated. Does your topic require current info, or will older sources work? It kind of depends on why you're looking for the paper. And of course, are links functional? The next letter is R for relevance. It's the importance of the information is for you. And so does the info relate to the topic that you're interested in? Does it answer the questions that you have? Who is the intended audience, and is that information appropriate? Would you be comfortable citing this source? And if the answer is no, then they get a little ding right there. The next thing is authority, is where the source of information. So who are the authors, publishers, and sponsors? Does the author have the credentials and the expertise to talk about said topic? Do they have a contact info on there? And what does the URL reveal about the source? So is it .gov, .edu, or is it like .net, .com? Usually .gov and .edus can be a little more trustworthy than compared to maybe .coms. The second A is accuracy, and that's for reliability and honesty of content. So where does the content come from? Is the information supported by evidence? Is it peer reviewed? Is it verifiable? Can you yourself, either with your own general knowledge that you already have, or go look for other sources, does that information stay consistent? And is it written in an unbiased tone? And then last but not least, P for purpose, the reason the info exists. So what is the purpose of the information? Or authors, sponsors, intentions, or purpose clear? Is it factual? Is it an opinion? Is it propaganda? Is their point of view objective and impartial? Are there any biases associated with that purpose? And here on the right in this figure, I have 3 different levels. So when y'all read papers, you can use this as a scale. For instance, where you look at currency, if the source is out of date or too old, maybe stop and find something that's more recent. Or if it doesn't reflect the latest advances or ideas, but it's not too old, you can just take caution. Or if it's most recent, you get the green light, and so forth. Does anybody have any questions before I go on?
I have a few.
Okay, what's up?
It was actually for the last section.
Okay.
So my question was, when you're starting your research, which kind of papers should you start with, and how far back?
Oh, that's a great question.
Oh, sorry.
Go ahead and finish. I'm so sorry, I got excited.
Which kind of papers should you start with? How far back should you look? And should you start with PubMed, or is there another database better for general research?
When you say general research, are you still talking about science, like healthcare sciences?
Yeah.
PubMed, I actually don't know why PubMed is the most popular, but it is. It's just the one that people mostly use. I have used Medline a couple of times, and there's another one. It starts with an O. I think it's Orchid, or cannot remember. But there's another one, and UAB also has access to it. PubMed is usually where you want to go. Now, if you're looking for something and you can't find it on PubMed, just go Google it. And if papers pop up, go see if they're available on another database. Once you find like the title of a paper, UAB has it set up where you can search for that paper and it'll tell you all the databases that have it. But PubMed is usually just a really good starting point. Now, what kind of papers? We're going to put a pause on that, and I'm going to come back to that towards the end of the presentation. Did you have a third question?
How far back should you look?
How far back should you look? That depends. There are classical papers that have been done in the 60s, 70s, 80s that people still refer to because they were just done so well, and they still just stand so true today. But for most part, it depends. If I'm looking for review papers, I might step it back and look within the last 10 years is usually my go-to. If I'm looking for primary resources, especially if I'm looking for evidence to use to write in my own papers, I stick within 5 years. If I'm having like zero luck in 5 years, I start dialing it back by 2 years. So I'll go to 7, 9, and whatnot. So usually about 10. But it just kind of depends on what you're looking for and why.
Okay.
Does that answer your question?
Yeah.
Okay.
One last question is, what is the DOI number? Like, what?
Oh, I forgot what that stands for. But the DOI number is just a identification number. So a lot of times that's what I record when, or I'll like copy and paste to my lab notebook. If you Google that or put it into PubMed, it immediately brings it up. It's like your driver license ID number.
Okay.
Mm-hmm.
Well, thank you. That was all.
Okay. All right. So now we're going to talk about how to read all these papers once we got them. So there's two ways I want to talk about reading papers. I'm going to talk about active reading, and then I'm going to talk about critical reading. And so active reading is a little more general. This can actually apply to anything that you read. It occurs in 4 phases. I got this from Teach Yourself How to Learn. It's an excellent book. I highly recommend it. It teaches you metacognitive ways on studying, and it's pretty legit. But from there, they do talk about active reading, and the 4 phases of active reading. You do a structural preview. The second, you do a content preview. Third, you do paraphrasing, where you put the content to your own words. And for the last one, you do engage reading, which is where you highlight or take notes. And so on the next slide, I'm going to talk about these more in detail. So here, the first phase, you preview the structure. Your brain will work more efficiently if it has the bigger picture and it's gathering the tiny little details and putting it in the bigger picture. So in other words, just like with puzzles, if you're putting a puzzle together, it is a lot easier to do a 1,000-piece puzzle when you have the cover of the box to look at and then put the tiny details together than it is to just put all those details with no reference whatsoever. Your brain works the same way with information. So with preview structure, this is how you get the big picture. And how to do that, you look for section headings, you look for bold print or italicized words, you look at all their figures and their legends, you read them, and then that gives your brain a kind of an idea of how they organize the information, when to expect what. And so when you go to the next phase where you preview the content, you can give yourself a reason to read by formulating questions that the reading can answer. Again, this allows your brain to work more effectively. No matter how old you are, your brain acts like a 4-year-old. It doesn't want to do what it's told to do just because you said so. You need to have a reason. You need to have motivation. And one way to do that with reading is you formulate questions based on the preview of the structure. For instance, like, why is this word, bold? Is it probably a definition? I know that the Teach Yourself How to Learn, they use, if you're reading in a chemistry chapter and you see both strong base and strong acids in bold, well, the first question you could ask is, what's the difference between the two? What are the similarities between the two? How do they interact with each other? So you write these questions down, these anticipated questions, so that you are looking for the answers as you're reading, and that'll keep you motivated in reading and not lose your spot or have to read something over and over and over again. All right, so for the third phase, you do paraphrasing, and this is the best way to paraphrase. So when you start reading, read only one paragraph at a time. Don't freak out. I know it's going to take a while. You actually don't really have, and just some real life advice, like, I don't do this for every single paragraph of every single thing that I read. Most of the time I started out that way, and then eventually I'll just read all the rest of the paragraphs without stopping, but still paraphrasing it in my mind. So to begin though, you read the first paragraph, then you stop and you put the information into your own words. Nobody's going to judge you. Use whatever language you want. Use whatever word, just whatever makes you know that you understand it to the best of your knowledge. Then you read the second paragraph and stop. You're going to paraphrase the second paragraph, however, also while trying to fold in the information from the first paragraph. Then you repeat it again with a third, fourth, and so on. And that will it will help you better understand the reading material as you're reading it. It will help you take notes, which leads me to my fourth phase, which is engaged reading. This is just where you highlight your note taking while you're reading. Paraphrasing, I usually like to put out my paraphrase as like bulleted notes on the side. Highlight important concepts. Y'all are all students. I'm sure y'all know how to use, y'all have y'all's own highlighter system. You can color code. I know that for me, like for concepts or words that I don't understand, I will actually put an orange box around it with my orange pen. Anything that's related to my project or homework assignment, I will highlight it in green. So you can do that way. So you can like scan through documents and find things that you need right then and there. However, word of caution, if your color code system consists of like 20 colors, it's probably going to be really hard to keep up with what they all mean. So like try to just pick the really important things that you want to highlight and just have a few colors to use. So that's all I have for active reading. Now we're going to get to critical reading. Critical reading is the thing that I would do for like journal clubs and maybe like for homework assignments. And so before I go on, does anybody have any questions? All right. So critical reading also has 4 phases. The first phase, you're going to skim the abstract, the figures, and the legends. Then you're going to go to phase 2. You're going to read the abstract in more detail and only using the figures as a guide if needed. Phase 3, you are going to read these in the order that they are listed. Introductions along with the references, the results sections along with the figures, the discussion section, and the methods last. And then for phase 4, this is where you sum up the paper in your own words, you review the paper if it's needed, and you can make your report for journal clubs. And I'm going to talk about each phase. So starting with phase 1, skimming the abstract and figures. So with the abstract, you want to read this first to get a sense of the subject area, the model, and the methods. It's just a rough guide in order to put the data into context. The second thing you're going to do during phase 1, you're going to go through the figures and the figure legends. You're going to read each figure line by line, and you're going to ask yourself, does the title of the figure legend explain the main question addressed in the figure? Is the data quantified? Do the figure legends give any clues of how it was quantified if it was? Were there positive or negative controls used? Or are they appropriate? Do you know? Do they explain it in the figure legends? They may or may not, just depends on the paper. And then explanation of how reproducible, like how many ends, how many replicates, how many technical replicates, biological replicates, and whatnot. There are actually other different questions you can ask in this in this section, but y'all can go to, I have the source down here. So if y'all want to read that whole article, I would definitely give it a good read through to get all the things in detail. But this is just the stuff that I picked out that I find that was most important. So basically for phase one, the objective is to just kind of make your own conclusions based on the figures so that you can see the story being told. And then you can compare your interpretation with the author's interpretation to see how consistent the interpretations are. Phase two, you're going to go through the abstract and figures in detail. You're going to review, you can review your notes from your figures and the figure legends that you read. You can read the abstract in detail and you ask yourself while you're reading, does the abstract explain the main question? Does it explain the main finding? Does it provide a rough sense of subject area, models, and technical approaches that was used? In this phase, the conclusions from a well-constructed paper should be easily interpreted from the figures and using the abstract as a guide only. It's to give you a better sense of the overall quality before reading the author's interpretation. Sound good? Okay. All right, so for phase three, doing the introduction part in detail. So quickly skim over what you already read through, which is your abstracts and your figures before going on to the remaining sections, but noting the structure of the argument as you go along. You want to read the introduction with the references, and the best way to do that is to either A, print the references as like starting as a new sheet of paper so that you can have a list of references beside the article that you have printed out, so you can like cross-reference at your desk instead of flipping through all the pages at once, or you can copy and paste the references into a Word document so you can control find when you get to references in the introduction that you're curious about or need to refer to. While you're reading the introduction is really just a general justification of research. It can be skimmed over, especially for me, I've read over several hundreds of kidney papers. Eventually the introduction starts sounding the same because there can be only so many ways you can describe the kidney. The more papers you read, the more likely you will be able to kind of more or less skim through the introduction rather than reading it word for word. However, if you haven't read a lot of papers in your field, you will probably be reading that word for word, and that's totally fine. That's how I start. That's how everybody starts. So while you're reading the introduction, you can ask yourself these questions. Is there an over-reliance on the citations of reviews? Papers should always use primary resources. It's okay if you find the primary resources through a review paper, but when you are writing a paper or when you find a paper, papers are just better quality when they actually have the primary sources cited instead of the review paper that they found, because that review paper may have 100 references. And if you really want to find that primary resource, you're going to have to go through all 100 references just to find it. It's a little aggravating. The next thing you can ask while you're reading the introduction is the background of the field explained well? What is the big question or subject area? Have authors identified a knowledge gap, and have they talked about how they're going to address that gap? So for the results sections, this is where you're going to cross-check your assessment of the figures, what you got from the figures, the figure legends, and the abstract. So your impressions from that with what the authors have talked about in the results and the discussion. So for each figure, several questions you can ask. Some I have highlighted here. Was the rationale explained in the text in the results sections for each experiment? Again, was positive, negative controls used? If it wasn't listed in the figure legends, it's most likely listed in the results. And are they appropriate? Do they explain if they were appropriate or not? Do you know just from general knowledge from being in a lab whether or not they're appropriate? Another question you can ask while you're reading results is, what is the author's interpretation and how does it compare to yours? And do authors consider alternative interpretations of the data? Can you think of alternative interpretations of the data? Of course, we always want our positive data to support our hypotheses, But sometimes just the evidence that we have collected can also signify something else is going on. Usually good quality papers will talk about these alternatives in the context of their paper and why or why not they think that the alternatives may also be true. And another thing you can look at is have they explained why they did the next experiment. And so after you go through the results, you also should ask yourself, could you follow the paper without consulting the supplemental figures? Some papers have supplemental figures because journals will only allow papers to have usually single digits, like six or seven figures in the actual article. So if you have more data to show that should go along with the paper, they put it into the supplemental section. A good quality paper should not rely on the supplemental data to make a clear case for what they're writing about. All right, so the next section would be the discussion. While you read through the discussion, these are some questions you can think about. Have the authors placed the results in the context of existing knowledge? Have they highlighted areas of agreement or disagreement? Have they acknowledged where alternative interpretations of their data is possible? Do they explain why they are not able to resolve any issues that they may have discussed? And do you agree with their conclusions? So from your impression off of what you read with the figures and the abstract before you read anything else, you should be able to come up with your own conclusions. It doesn't mean that you'll conclude everything the author has, but you may conclude some of it, and do your conclusions coincide with theirs? Usually if a paper is written really well, the better it is written, the more likely your conclusions should be really similar to theirs. And then last but not least, you go through the methods and details. The big question is, when you read through the methods, do they provide enough detail where you could repeat the experiment on your own? If not, what missing information is there? Should that information have been included? Have they used appropriate terminology or slang? In other words, instead of saying separated by centrifugation, they just said spun down. That's okay if you're talking in lab meeting or informally with someone, but a good paper will not use that slang terminology. Another thing you can ask while reading the methods is, have they indicated where they purchased or obtained their reagents, their assays, even their animals? Have they explained how the data was quantified? Did they provide justifications for the models used or for the statistics used? It shouldn't be any surprise to anyone that there are several different, like for me, I keep going back to animal research, there's several different animal models. There's many different statistical methods that you can use. Obviously, we don't have all the time or money to do everything. So you have to pick your best choice. Usually in the methods, you would like to address why you think it was the best choice, if it's a really good paper. So for the last phase, after you're done reading through everything in detail, sit back, reflect on it. Do you trust the paper? Like, would you put your name on that paper? Do you feel confident that they did the best they could do? What did you like about the paper? If you do not concur with the author's conclusions or found that the data quality is unsatisfactory, what would you conclude about what was learned, and what would you have done differently to make it better? How would you summarize the paper in 3 to 4 sentences? Basically, like, to really put it bluntly, a really good paper, the key objective is to be fully transparent. You should be able to look at the data, draw conclusions, and check those conclusions with the authors, and they should match. If you draw different conclusions or you're unable to, it may be a sign the paper was not written or presented optimally. It doesn't necessarily mean they didn't do the experiments right, or their ideas were bad, or their critical thinking was flawed. It just may just not have been written optimally. This could be due to it just wasn't written properly or optimally by the authors, or it may be due in part because of journal restrictions. You do have word restrictions. Again, I've already mentioned about figures. You can't put every figure that you made. that's pertaining to that study. You can only put maybe 6. So you have to take that into account as well. But overall, you should be able to draw your own conclusions, and it should be comparative to the authors. All right, this is my favorite part. I'm super excited. This is what I really wanted to get to. I want to show you how to make databases, because once you get all that information, how in the world do you keep up with it? Because if you're like me, my brain's not big enough to keep it all in there. Okay, it depends on what the information is for and what you're going to do with it. So you can create your own databases to keep up with and organize your info. But the purpose of the reading can guide you on what kind of database to make. So there are different ways you can, different databases you can use when you want to insert references into like a Word document. Like, so if you're writing your own paper and you're looking for references, there are different programs that can insert into the Word doc directly. Things like EndNote. I have a favorite one now that's free. I can't wait to show y'all. There's another one I called method centric, but it's anything kind of like centric. So something that you're looking at the paper for a very specific bit of information that you really don't even need to read the whole paper to get it. Most of the time, that's when you're looking for different methods. And then of course, general knowledge. This goes back to Nilu, I think, was asking about what kind of papers you usually start with. I usually start with review papers. If you're just trying to read for general knowledge, I try to find all the review papers I can about it, because those are great sources of primary sources. A review paper should not use other reviews as reference papers. Kind of defeats the purpose of a review paper. It should have all of its references should be primary sources. So that's why I usually like to start with review papers when I'm looking up stuff, just to get for my general knowledge. I'm going to check the chat and see what I got. I think I got 2 people. Oh, digital objective identifiers, DOI, FYI. But anyway, so PaperPal. There are different programs like EndNote where you can use to insert the references into your Word docs. EndNote is not free. It is through UAB and other universities. So you can check with your university and see if they have an account where you can use EndNote through them. However, that does present a problem. For instance, I did my PhD at a different university than UAB. UMC did have EndNote, but that isn't always the case when you move on to your next spot in your career. So there are other ways or other databases you can use. One is PaperPal. It's free, so it can go wherever you go. It's a web-based commercial reference management software. It works with Google Scholars and Google Docs really, really well. It can be implemented as a Chrome browser extension. So you can actually, even on PubMed, you can Google whatever you want, keywords. It'll give you all these lists of papers. There is a little P that will pop up once you have that browser extension installed, and you can click on it, and it automatically adds that paper to your account. It is super streamlined. It's wonderful. And the best part, it's free. And so I wanted to show y'all what does it look like. So if you go set up your account with them, again, it's free. This is my PaperPal. So you can make your own folders. This is just how I organize my folders for my projects. You can also have these things called labels. The one that I actually use frequently is the README. So a lot of times when I add papers, when I haven't read them yet, I put the README on here to let me know that I haven't read this paper yet and I should read it pretty soon. But you can do whatever labels you like. You can search for keywords up here. When you add a paper to PaperPal, for instance, let's go here. Okay, does this one have? Nope. This one has an attached file. So let's look at this one. So this is a paper that I found to look at for my project. If there's a PDF already available, like if the full text is available through PubMed, it automatically attaches that PDF to the paper in PaperPal. If it does not, and you can get the PDF through alternative sources, you can go back to that paper and paper pile and attach it yourself. For instance, here you can click the paper clip. This one didn't have a PDF with it. However, I can attach my own PDF to it. So if you can find the PDF of the full text, you can go back and attach it. Now, when you do that, you can actually read your papers on paper pile. You can highlight if you want to. You can take notes on the side. Usually, every time I read a paper, I put the big main takeaways from that paper, or like why I read that paper and the stuff that I found that's in the context of that. I usually also have a little box here that says whether or not it's downloaded by me or was sent to be by my PI, and what date I downloaded it, and that sort of thing. And so if you go on paperpile.com, you can play with it. Again, it's free and super easy to use. So let me go back to here. The next thing I want to talk about is method-centric databases. So a lot of times you're like, for instance, again, using my experiences. So in a lab, like in a research lab, usually if you're trying to do a technique with like a particular tissue type where the technique has been around for a while, it's been used on all these other things, but not necessarily on the way you want to use it. What you can do is you can go, again, use the method keywords. For instance, if you're looking for a particular stain, so like oil red stains neutral fats. I used to do obesity research. So I wanted to see, how do you stain kidneys with oil red? I wanted to see whether or not are they tissue specific? Are like protocols based off tissue specificity? Is it species dependent? Another thing you can do, or what a lot of people do, is Western blots. If you've never done a Western blot before, yeah, they hurt, but they are the gold standard of a lot of wet lab research. And every step can be basically personalized, can be tweaked. So like, for instance, antibody dilutions, how much tissue should you use to digest? Should it be 1 milligram, 10 milligrams? Is that species dependent? Other things you can do, if you're doing in vitro work, looking up the cell size, or how many numbers of cells you can isolate from a certain type of cell. And are these, again, species specific? And even in the same species, you have different strains. And so stuff can be strain specific. So it's a lot of information. What I like to do is, for instance, I was trying to figure out what filter size to use to isolate cells from parts of the kidney in rats. And I wanted to see what have other people done previously in the literature. And so what I do is I will Google podocyte isolation in mice or rats or whatever. A bunch of papers will pop up. The ones that I found helpful, what I do is I will copy and paste the title of that paper, the authors of that paper, and the affiliation, and then I go straight to the methods and look for the very specific information that I would like. So usually I look for what species they used, what pore size they used, and then I go on, oops, my bad. Then I go on to the next paper and repeat the same thing. And that way you can get all of that information through several papers and have it in one location, and it'll help you make a decision on what type of protocol you can use. And that way, when your PI comes up to you and be like, well, why do you think that? And you can be confident that you definitely came up with the right answer. But if you can't prove it, then it's not really worth a lot. I usually just pull up these kind of documents that I have and be like, because all of these people did it this way, and this is how I figured out this is the best way to do it. Now, for my favorite thing. This is an Excel file. How do you keep up with all of the papers that you read? I have made an Excel sheet where I can organize it by whatever I would like. So for instance, disease, model type, technology used, the senior author, the first author, by dates, by databases, whether it's public or private. Each one of these slicers is a column where I have all this information for from those papers put in here. Let me go to the next one. So before I show you how to make that, let me show you how I use it. So when I'm reading a paper, I usually download the paper to a paper file and put the README on it in case I don't have time to read it right then and there. But when I am ready to read it, I open up that paper and then I put when it was downloaded, or what was it downloaded by me, or was it sent to me by my PI when I downloaded it, what's the main takeaways of the paper. And if there was a specific reason why I was looking for that paper, for instance, again, if it's method centric, you can write, I was looking for how to do this particular method or something for this particular project, or it was a homework assignment for class. I add the logistics of the paper, so like the authors, the year it was published, to that personalized Excel sheet. And as I'm reading the paper, I fill in the rest of the fields. I add a date read to the Excel sheet and then remove the README from tag from that paper. And so I think we have a little bit of time. I want to at least show y'all, make sure this link works. This is a YouTube video.
So I've been building this over the past.
This dude shows you exactly how to make this Excel sheet. It takes less than 10 minutes to do, so don't be intimidated by all the pretty colors and the pretty slicers. It's really, really easy.
It's 3 years now, like 2 years now. And I'll fill this in as I'm reading a paper. So first and foremost, just be aware that
I didn't start it. I was watching this before I hopped on with y'all.
screens. So,
all right, so this is how it starts. This is how simple it is. I just wanted to show you all the beginning of it. You can have the title of your paper as a column, the author of the paper, whatever their names are, year, paper type, then the main topic, subtopic, method, summary, and then you keep any other column that you would like to have, you can add it here. And when you have that all filled in, you're going to highlight and make it into a table, and you're just going to add what these things are called splicers. And so I'll let y'all finish this video on y'all's own time, because I want to make sure, because I think that's the last bit of information I had to share. So the only other thing I had to share was, so this is my database, my personalized database. I was looking for publicly available data sets to analyze for different kidney diseases. You can see up here, there we go. So I have the different diseases like acute kidney disease, chronic kidney disease, end stage, normal, polycystic. Was it in humans, mouse, rat? So basically what happens is when you make a different column, all the information that you put in automatically, if it's not already been made, it's made up here. And the way you use this is, let's say for instance, my mentor came to me and she's like, hey, are there any databases where it's chronic kidney disease and rats? So you can come over here in your Excel sheet, you click chronic kidney disease, and it pulls up all of the chronic kidney disease databases. Oh, I don't have one for rats. As you can see, it is faded out, but I do have them for human. So I can click that and it'll just show all of the chronic kidney disease databases that are just human. So this is a way you can keep up with and find your papers, if that makes sense, in a much quicker way than trying to like keep them all in a notebook. And with that, that's all I had. Does anybody have any questions? Or did I talk too fast and lose everybody? That could also be a thing.
I don't have a question, but I just want to say thank you for the database. I didn't know that that was a thing.
Okay, to go ahead and be honest, I didn't know that was a thing either until July of 2020. I saw it as a tweet from like, I think her name is on Twitter, Thoughts of a PhD. She had like it all listed and had that YouTube video, and it has changed my life. So I wanted to make sure y'all had that video and use that. If you start using that now, future you will thank you later.
All right. Keegan, Haley, any last comments from you?
Oh, I just wanted to say thanks. I thought that was great.
Thank you. Good.
I also wanted to say thank you.
I wanted to cut all the fluff and give y'all everything that's usable because we ain't got time for fluff.
That was great. So again, just to remind folks, we will put this up on our YouTube channel. So if you joined us late, we'll have the whole thing up there on our YouTube channel. Thanks, everyone.